Okay, welcome back to what is episode 15 of Ag Inspired Conversations this morning, and my name is Tommy Heffernan, and the concept is fairly simple, and we're getting closer to it every time. Interesting conversations with people doing inspiring things in agriculture. And my next guest, I'm going across the Irish Sea again up to Scotland to talk to Dr. Philip Skuce, who has had a very interesting career, and he has a common interest to me, and that's around parasites. Now, he knows a lot more than I do about parasites, so we're going to talk a little bit about that. First and foremost, good morning, Philip, and thanks for joining me.
Good morning, Tommy, and greetings from sunny Edinburgh. That's not usual. Yeah, I mean, it's a bit surreal to see you for real. I've seen clips of your Ag Inspire conversations before, and it's a bit surreal to see that. And I'm also slightly humbled by some of the people you've spoken to. I mean, you've got all sorts of very interesting people doing very interesting things, and you've got life coaches and all sorts. I wouldn't claim to be that, but yeah, looking forward to our chat. And normally we'd have a pint of something black between us, maybe if times were normal.
But it's interesting, Philip. You say that this is just something that I noticed. Everybody I ask, I think they're all extraordinary in the various, whether they're doing for their farming and research or whatever. But a lot of people have that feeling of, oh my God, what am I going to offer this conversation? So it's an interesting thing, and I think, do we all suffer? I know it's probably a strange place to start. Do we all suffer a little bit from imposter syndrome sometimes? Do we?
Oh, I think so. I've still got that many decades into my career. Yeah, I would totally agree. But it's interesting how our paths crossed, actually, because I think you'd spotted some of the things I'd done online, based up in Scotland here, maybe, surprised and pleased that people across the Irish Sea could see what we do. But I also spotted you. So we've never met in person, but I have seen some of the things you've done for farmers and with farmers, and that sort of intrigued me, because I think sense communication is a key part of what we all try to do.
It's very important. So, okay, let's find out a bit of your own history. I know you studied in Queen's University. You're in Moredun Research Centre in Scotland now. But tell people, who's Philip Skuce, Dr. Philip Skuce, and your path into research and agriculture.
Yeah, that's an interesting one, Tommy. Slightly convoluted as well, I guess. You can tell from my accent I'm Northern Irish by birth, but the Skuce family home is actually West Cork. So I still have family there, and it's a lovely part of the world, and we like to get there when we can, and hope to get there again soon. But yeah, interestingly, there were no scientists as far as I knew in the family. In fact, my dad was a Methodist minister. Okay. So travelled around the country. So when you asked me where I was from earlier, that's a good question, because we moved around a fair bit, mostly in the north, but a little bit in the south, Dublin and Cavan. So yeah, no scientists in the family. But then, like, buses too came along at once, because I'm a twin. Okay. My twin brother's a scientist as well. Wow. Folks in Ireland might even know him, because he works on bovine tuberculosis in the vet labs in Stormont. So I know for a fact he's been across the country doing various talks in north and south. So if this all goes wrong, I'll pretend I'm him. But yeah, so you get two scientists at once, and yeah, I guess our paths have kind of almost converged, and we did work together at one point, and I'll mention that a wee bit. But yeah, I just kind of enjoyed life sciences at school, and biology in particular. I was never the sort of numerate hardcore physics and chemistry. I always liked biology, and went on to study animal science, or zoology as it was called in the day, in Queen's in the 1980s. And I think a couple of previous speakers, John and Des, both mentioned the troubles, and yeah, I guess we grew up right in the middle of that. So school and university was an interesting experience. But I really enjoyed my time at Queen's, and I was very lucky then, because at that time we had 3 or 4 really eminent parasitologists in the department. Folks might know some of them: Dave Halton, Bob Hanna, who actually is a pathologist working at BSD now, and Ian Fairweather, who went on to become my PhD supervisor. And they were all really influential. I got intrigued with parasites there and then, and that became what I did my PhD on.
It's something that's common, Philip, right? That when you see people who are interested in parasitology, it's a real passion and interest. Now, if you say to somebody removed out of parasitology, how do you get interested? And I see farmers, and my very first guest, Bruce Thompson, is fascinated by them. How do you get interested in something like parasites? What is it? Is the life cycles of them? Is it what was the thing that intrigued you about them?
No, I mean, it's a fascinating discussion actually, because farmers just want to kill the damn things, and I can understand that. I'm fascinated by how they work and how they survive, and there's all sorts of evolution in action. How do they become resistant to things? How can you vaccinate against them? That sort of thing. But I think the public reaction is usually revulsion. How on earth you spend your life doing that? And even sampling, we all spend days picking up fecal samples and say, or looking for snails, and why don't you get a real job? But I mean, but I just find them fascinating. It's how they work, how complex they are. I mean, people think they're primitive. They're anything but primitive. They're highly adapted and highly evolved, big genomes, complicated organisms. They can live in the soil, on the ground, in an animal, in different organ systems, in water, in different, snails and deer and rabbits, you name it. They can pretty much go in anything. So I think it was that sort of fascination, really, and how do we sustainably control them? What are the sort of weak points, and what could we do? And I mean, again, the veterinary side is important, and the big thing, as you know better than me, that farmers spend a lot of time and money and work trying to control. I mean, they're major human parasites too. They just happen to be in tropical countries that don't have a lot of money to deal with them. But it was just that spectrum across the board. They're everywhere. They're ubiquitous. They're successful. They're fascinating. So I think I do need to get out more, but I'm not allowed to.
No, it is a fascinating subject. Like I have a small interest in lots of different things, but I can see how parasitology becomes. You can get so immersed in it. And just one thing I read about, and I was actually no, I didn't read it. I was talking at a bar one night later than I should have been with a guy, and he was talking to me about the origins of ivermectin and how that was discovered.
Mm-hmm.
But what I found most fascinating was we think about ivermectin for animals, but that's been revolutionary for people. Yeah. When we talk about parasites in animals, let's just go to people just for a minute. It's one of the big global challenges, isn't it, around health, Philip?
Yeah, and I mean, I think what says a lot is actually that most of the parasitic diseases are described as neglected tropical diseases. Okay. So they are neglected by governments and funding agencies, and to some extent, large pharmaceutical companies who produce things like ivermectin down the line, but there's no real market for that because who's going to pay for it? And so that story, I mean, you mentioned ivermectin, and it's a really interesting Irish connection there, as you well know. The inventor or discoverer was a very famous Irish parasitologist, Professor William Campbell, a Northern Irishman. So another Ulsterman done good.
Yeah.
But he and Japanese colleagues discovered ivermectin, would you believe, in a fungus growing or a mold growing on a Japanese golf course. So that's my interest in golf covered as well. But they went on to do fascinating things with that.
Just tell people about that discovery, because that was another thing, that it was almost like an accidental discovery.
Yep. Like some of the best scientific discoveries are. It was pretty serendipitous. It was drug company Merck back in the day. I mean, they do a lot of screening. And that's how you typically find drugs. You look for biological activity, and this particular mold had a compound in it, or a group of compounds in, that had a remarkable activity against nematode parasites in particular. And it was used then to treat as the frontline treatment, and still is, for river blindness, which is a disease called Oncochiasis, which is a big problem in the tropics, as we said. It's got a fascinating life cycle too. It's got a vector, a black fly. So there's a river connection, there's a blindness connection, because the larval stages have a predilection for the optic nerve. And it's just a fascinating story. And it's timely because I think Professor Campbell, he got a Nobel Prize for that a couple of years ago, and that in itself is really off the charts, because I don't think any Irish scientist has ever got a Nobel Prize for medicine, and he did. And he's a very humble man, and I would encourage any of your listeners, if there's anybody still listening, to have a look online, because his Nobel Prize lecture is online, and you'll see just how humble and nice a man he is. And actually the Irish, I think it was the Royal Society of Ireland, he's just produced a book, I think it's around his 90th birthday. Wow. And there's a very nice sort of conversation online with RSI and a couple of Irish scientists. And again, he displays his humanity, and the whole story is fascinating, and encourage people to buy the book if you can get it, actually, Catching the Worm. It's fascinating. I mean, ivermectin really has revolutionized how we do things, or how you do things. And a whole family of chemicals like that have arisen. And they've revolutionized livestock, certainly how we treat parasites in livestock. I mean, there are some off-target things, and there's becoming an increasing interest in sort of ivermectin and in the environment and sorts of things like that. I mean, they're used in fish farms, they're used to treat horses. Just, I mean, it's fascinating. But it's just the whole story of where do these drugs come from and what motivates companies to produce them. We don't have that many. I mean, farmers think we've got lots of chemicals and there'll be another one along in a minute if this one doesn't work, but we're not there. And it's really communicating those messages. That's kind of key. But I was very lucky at Queen's that was a good time and we had those people there. And so that was a springboard for a sort of a career in parasitology, which I would have liked, and I'm very fortunate to still be here, but I probably couldn't have planned. I think that was back in the day when you just weren't quite sure how things would work out.
And you went from Queen's to Moredun Research Centre then?
No, it didn't actually. I had a wee sojourn in between. I was lucky enough to get a fellowship from the Medical Research Council to look at the human cousin of the liver fluke called Schistosomes or Schistosomiasis, Bilharzia. And I hate to say it, they're even more fascinating than liver fluke because they're separate sexes. There's males and female schistosomes. They live in the blood system that drains the intestine or the bladder, and they cause a really nasty tropical disease called Bilharzia. I mean, they really are fascinating. If you take the males and females apart, you can do that in a petri dish. The female will regress and become immature again. And if you even if you cut the male into pieces and put the pieces of the male in a petri dish with the female, they will still pair with the female and she will mature where they're in contact. So I mean, absolutely fascinating because it's the egg-laying habit of the adults that causes the disease because the eggs get swept into the spleen and the liver and cause a really nasty disease. So that was fascinating. That was in a clinical department of medicine at the Royal Victoria Hospital in Belfast. We were looking for targets for drugs really at that time. So we were investigating the nervous system of these parasites, which was really just starting to become a very interesting topic. They're full of hormones and peptides that are sort of precursors for things that we have in our systems too, in the pancreas and various other organs. So that was fascinating, looking at the nervous system of all sorts of stages of schistosome parasites. So
people will see that as incredible detail, Philip. But without that detail, we can't control these parasites. So if I'm a farmer and I have an anti-parasitic product, the reason I have it is because of the detailed research that goes into understanding what people think a liver fluke or a nematode, a worm. It's a simple organism, but it's not, is it?
No, I would argue that they're not simple. And the more we learn and the more technology moves on, sort of sequencing capacity we have now to sequence genomes and genes, and that's fascinating because they use those methods to amplify DNA, like PCR we've all heard about with COVID, and there are methods for detecting, so improved diagnostics. So that all comes full circle. But you're right, it's a combination of, I guess there's academic work and to some extent into understanding, the genome of an organism is its instruction book. How does that make that organism work? How can we intervene in that? Drug companies take a slightly different. They're more, as I say, screening. The very few drugs have ever made the market as a result of a think tank, somebody sitting and thinking hard. They tend to be done through screening. So as a result of that, quite often don't know how they work. And that becomes the academic challenge. And how does that work? And then how does the parasite get around that? How do they become resistant? Why is that treatment not working anymore? And that's the really fascinating bit where the two kind of come together.
I know I keep interrupting, and you haven't got to Moredun Research Centre yet. But when
I'm still in Belfast.
When people hear about genome, though, it is something, and I went through years of college and everything, and there's lots of words that you hear and you're qualified, and you nod that and then it gets too much nodding.
I nod as well. Don't worry.
And you can't say, but just explain to people, because we will talk about the genome again. It's coming up time and time again with genetic editing and everything. But what is the genome, and how mapping it, whether it's the parasite or anything, how important it is from a scientific perspective? Can you just go into?
Yeah, I think it's a new technology really, although it's advancing very, very quickly. I can remember back in the day, and I maybe should mention how I got into this side of things, but it's a core discipline now, molecular biology, I guess you'd call it, or genetics, genomics. But it's understanding how animals reproduce and breed. But the genome is the instruction book. It's like your car manual. It tells you, in chromosomes and genes on chromosomes, how do the life stages work? How do they digest their food? How do they reproduce? And it's all about interpreting genomes, but the technology is fascinating as to how quickly you can sequence genomes now, and the computational skill that goes into that, and the ability to annotate to know where one gene stops and another one starts, and where's the switches, what switches that on, what switches that off, and then get into things like drug resistance, where you're looking for spelling mistakes, so the gene's gone wrong, what's wrong with it, or maybe a parasite survives the treatment, so how's it done that? And there's a classic case with white drenches that people will know about, the benzimidazole group. They're the best, most understood group, I guess. We know for a fact that they interact with tubulin, which is a protein in parasite cells. We've got tubulin in our cells as well. Thankfully, it's a bit different. Otherwise, we'd have trouble with white drenches. But we know for a fact that those drugs bind to tubulin and stop various things happening in cells, including animals, so the parasite's feeding. And a single spelling mistake, one single base in a long gene is responsible for that resistance, and it's inherited, and we can track it and trace it and see what happens when you treat and when you don't. So that's where the science is coming full circle. So it's giving us the ability to do detection, to do vaccine studies. I'll mention that a wee bit in a minute if I ever leave Belfast. But
Belfast.
I was very lucky actually, because as part of the fellowship at the hospital, I got a trip to Germany for a year, and it was to Dusseldorf, which had a very famous institute for genetics at the time, and I had O-level German, so I was able to get by. But I learned a whole load of new words. But that was really interesting, because molecular biology was literally just starting then. You had no such thing as PCR machines. They built their own. It was at that sort of level, but it was a really interesting grounding right at the start, and I was able to take that back and got a job actually in Belfast in the vet labs working in the same lab as my brother, which caused all sorts of problems.
I can imagine. Are you identical twins?
Well, I think we're very close. I'll send you a picture later and see if you can tell the difference. But we are very close, and I think the fact our aptitudes and interests are absolutely identical. Okay. I'm not altogether sure of technically whether we are or not, but we are very alike.
Okay.
Extremely, yeah. So I was able to bring that sort of embryonic molecular biology back with me to BSD, and I worked for a couple of years on a vaccine for red water fever, Babesia, which is a protozoan parasite, which is fascinating as well, although I do prefer the helminths. Okay. But then my brother actually spotted an advert for a job at Moredun. To be honest, and I hope my colleagues aren't listening, I hadn't really heard of Moredun, because we were kind of trematode people. We were flatworm people, but they were more roundworm, nematode people. So he said, oh, there's a parasitology job at Moredun. Like, he knew people working on paratuberculosis. So I applied for the job, not expecting to get it. And it was only for a year. I thought, well, it'll be a nice place to work. And so it proved. And I'm still there. This is 25 years on, would you believe?
And are you head of research?
Oh, God, no. Definitely my colleagues won't be listening to this. I run a very small sort of fluke research team, okay, with the students and one research assistant in amongst a bigger parasitology group. Okay. So we don't really have a head of parasitology as such. We kind of spread the load. I've got some very good colleagues, Dave Bartley, Fiona Kenyon, people will know from the work they've done on drug resistance and targeted selective treatment. You might have heard of that concept.
I've heard that. I was going to ask you about that.
Yeah,
I was going to get into the parasites with you. Sorry, I just have a habit of interrupting people when they talk, when they say something fascinating. You used the word there that I probably would have never heard before, and maybe never again. They were nematode people, and we were helminth people.
I probably should have defined. Well, we were kind of trematode people. At Queen's was very strong on flukes, flatworms, and tape. So that's right, flukes and tapeworms. The tapeworms are less of an issue for certainly for veterinary parasitology. They're a bit more cosmetic, but nematodes are a big problem. I would have studied them a bit as an undergrad, but hadn't done much since. I wasn't really expecting to get the job, but I guess I had transferable skills, having worked a bit in molecular biology. So the project I was involved in at that time was a really fascinating early stages of what went on to become Barbervax. You'll have heard of Homunculus, which is a blood-feeding nematode, which is arguably the most important nematode of small ruminants in the world. Big problem in sheep and goats, even in this part of the world. But
it's a growing concern for farmers in the ground who wouldn't have heard of it. We're seeing more incidents of it.
Yeah, I think we would see that too, actually. And we don't tend to see clinical signs of Haemoncosis in Scotland because it's a bit cool up here, but certainly south of England would see that as a major problem, and it seems to be a problem in goats in Wales. So you see sporadic appearance of it. I mean, we did a little project a few years back where we sampled fecal samples that farmers had sent in from across the UK, and we were able to look using molecular methods, DNA-based methods. We could detect the Haemonchus eggs in the fecal sample, and you could tell there was a kind of gradient south to north, but there's about 50% of the farms in the south had Haemonchus eggs in their fecal samples, so it was circulating, although you didn't always have clinical disease. But in the north, it kind of tapered off a bit, but it wasn't. It was there, and we found it in Scotland in about 20% of the samples we were testing. So it's out there. It just wasn't surviving our winters or whatever. But I think the climate is slowly changing, and that might change because we do on occasion see Haemonchus now. But it has a real propensity. It's got a big genome. It's a complicated beastie, and it has a real propensity to become resistant to things. It's resistant to everything we've thrown at it so far, including some of the new actives that people might have heard of, the orange drenches, monipantel. Yeah. So it's resistant to white drenches, levamisole, and most, if not all, of the macrocyclic lactones because it's genetically complicated and it's got ways of getting around things. If we treat it with a chemical, that's just another insult, and it will get around that in time if we keep doing that. So that's when the thought process has to come in. Well, how do we manage this better? How do we detect it better? And that's the research, I guess. But we were trying to vaccinate, and I mean, one of the things that's key about Haemonchus is it's a blood feeder, and there's an opportunity to see if we can attack it from the inside out, and it's likened to kind of Trojan horse thing. So what we were able to do is extract proteins from its gut. We can actually dissect the gut out and extract proteins from the gut, and when you vaccinate sheep with those, they mount an antibody response. And when they get infected with Haemonchus, and it takes in blood with antibodies, it kills it from the inside out, and it works beautifully. That's what I would describe as a native protein preparation. So that's a protein preparation detergent extract of the parasite itself. And the tiny little things about the size of an eyelash. So how are you ever going to make enough of that? So we had in principle, a vaccine that worked, but how are you ever going to manufacture that? I mean, so that's where we were, and that's where molecular biology came in. Could we use the anti-sera from these protected animals to detect what is it they're reacting to? What's protective about that fraction? And that's what we did, and that was my job, and that worked really well. So over the next few years, we were able to literally name all the parts, all the components of this Haemonchus vaccine protein preparation. A few publications out of it, nice piece of work. The next job was, could you synthesize those artificially? And we have a number of options there. That's to use another technical term, that's recombinant.
Okay.
So you're actually putting a gene into a vector and getting it to express the protein for you. There are a few options. Bacteria would be the obvious one. That's the easiest. Yeast, quite a good one. We're quite good at fermenting things in Ireland and Scotland. You can even use insect cells or mammalian cells in culture. So we tried all of those, and we could make pretty much all of these antigens. They all had the right protein sequence. We could prove that. None of them protected, either on their own or in combination. And that was disappointing, but that's been the way with vaccine work. And it's because the recombinant or artificial proteins we made never had the right shape, because that's what the immune response reacts to, is very fine shape.
Making of a vaccine for your parasites.
Yep.
Before we get into that, because I think it is very fascinating and people will be interested in that, let's move out to the big picture for a second, right? Let's move out to warming climate. I say to farmers, one of the biggest limiting factors in our grass-based systems over the next 5 to 10 years is parasites. I'm seeing it on farms. We've 3 active agents in cattle, we've 4 in sheep here, 5 in the UK, and we're getting resistance issues, we're getting more issues with parasites. Okay, so I think, and I know you might agree with me, that this is a big challenge. So you've gone into a lot of detail there, but the reason for that detail is parasites are one of our biggest challenges. Am I correct in saying that?
You're absolutely right, Tommy. I wouldn't disagree with anything you've said there. They spend a lot of their life history outside, whether that's as a roundworm, larval stages, or especially flukes, I would say, because they go through a little mud snail intermediate host. So they spend a lot of their time outside. And a lot of the components of their life cycles are dictated. The speed at which they happen, the success and the efficiency of that is driven by the weather, and weather over time is climate. And we've seen trends of warming and wetting, and those are situations that parasites love. That's why they're a big problem in the tropics, warm and wet. That's what parasites like. So they survive better over winter. We're talking about Homuncus there, and we've got evidence at Moredun that we're starting to see over winter survival. That was something we didn't think would happen. With fluke, we're seeing it out of season. Then back in the day, you could be quite prescriptive. You knew exactly what fluke was going to do. There were certain things would happen at certain times of year, but I think that's slipping now, and we need to be aware of that. And I mean, we encourage farmers to do much more monitoring and testing, and so that they know what's happening and what's normal and what's not normal. But you're absolutely right. We'll see that across the board with all the species. I mean, they're all affected to some extent. The speed at which they, as I say, for fluke, it's how quickly do the eggs develop and hatch? How quickly does it go through the snails? How successful is that? How many snails are there? You know, Ireland and in the west of Scotland are famous for being lovely and mild and wet, and that's perfect for snail habitat and worms. So I think we will, as you say, we're interested in the sort of impacts of climate change on parasites and pathogens. And also, I mean, something we might come back to later, these are major production limiting diseases, and there's a big push to try and improve efficiencies and reduce the number of livestock and all this sort of thing, and make them more efficient at least. And this is a big break on that. So there's two sides to that argument. We're trying to reduce disease as well, to reduce emissions and improve efficiencies. So that's the sort of political side of it. But you're absolutely right.
We're going to have to tear up the rule book when it comes to parasite control, or at least change it, because it was so easy. I had a parasite problem, I dosed, and I was able to control my production from that perspective. Now legislation is changing. I'm going to need to use less anthelmintics, and there's complete reasoning for that. So we talked about testing and diagnostics and understanding the risk. Just before I get to vaccinations, any thoughts around species of swards and parasite control?
I mean, that's not really my area of expertise, Tommy. You've caught me on one, but yeah, I mean, there's an increasing interest in Scotland and Ireland are pretty green. Well, Ireland's very green, and our livestock systems are grass-based, and I think we should stick to what we're good at. I think we're good at that, but we need to make more use of that and not rely on chemicals as much as we have. I mean, you're absolutely right. Farmers treat with a product at a certain time of year because that's what they've always done. That isn't sustainable. We know that now, and that we have to be a bit more integrated in how we control parasites. We have to use other options. I mean, with fluke, you've got some options around like drainage and fencing and things, management options. I think you're right. We need to look at, where animals are, rotational grazing and combined sheep and cattle grazing at the right time to do the right job. So it's understanding the holistic kind of approach. And there's also a big push when we've talked about climate change, but there's an equal push coming down the line on biodiversity loss. And we can't have, and the governments won't stand for, sort of monocultures of nothing, whether that's arable or livestock, just nothing. So we need to think about that as well. And livestock are great conservation grazers, and it's trying to.
I want to get to that at the end, because I know you're involved in the project. Just before we sort of leave the parasites, right? So we've established resistance an issue, and it's a very hard sell to farmers. But I am trying to tell them that unfortunately parasite control is going to get a little bit more complicated, whether we like it or not. But there is some hope in the horizon, and I think you mentioned it in the form of vaccinations. Where are we with vaccines for our nematodes, which are roundworms, and our liver fluke? Is it something that's in the near future? Is there practical applications to it? Where are we in that space, do you think?
Yeah, I mean, just to complete the story on Homuncus, actually, is kind of where we'd left it. Having done all that nice work on naming all the parts and trying to produce the recombinants, none of them working because of this three-dimensional, they just don't have the right shape to be protective. My colleagues David Smith and George Newlands worked on with that, and I kind of left that project a little bit behind me. My work was done in that regard, and I moved on to the genetics, molecular genetics of drug resistance. But I'll just finish George and David's story because it's got a happy ending. Okay. There was a sort of light bulb moment at the end of that period. using a very fancy electron microscope, they could actually physically see the vaccine complex they were using, so the proteins we were characterizing. And when we saw it, we were like, good grief, that's complicated. It's in shape. We'll never produce that synthetically. So the question arose, well, could we ever produce enough of it from parasite material? And one key part of that was, well, if we dilute down the antigen we've got, how many doses could we get out of it? And it worked at about 1,000-fold less than we thought. So there's 1,000-fold amplification already. And then I thought, well, could we produce more homunculus to extract it from? And that became feasible as well through collaboration with Australia, where homunculus is a big problem. And what they were doing there was giving sheep a very small dose of homunculus, just enough to set up infection, and they were going to the abattoir anyway. So harvesting homunculus from those sheep at the abattoir and making the vaccine from that. So it's a protein preparation extracted from real parasites, and it works a treat, and it's been commercialized and is now selling like hotcakes in Australia because they've got all sorts of drug resistance problems, and homunculus is their #1 parasite. So I'd encourage anybody to have a wee look at the BarberVax website, see the story. And there's a story, there's a really interesting science story. We've gone full circle and gone native, to use the technical term. So there's a vaccine for homunculus, and it is available in the UK and I think in Ireland under a special veterinary license. The other parasites are more challenging in a way because, with the exception of liver fluke, they're not blood feeders, and we can't use that same approach. So we have to try and really understand the immune response and how sheep and cattle become immune naturally, and try and build on that. So there's a lot of work going on in that area. And I think it will go down the recombinant route, the DNA sort of artificial synthesis route, because drug companies or whoever might take this on commercially, BarberVax is basically done in-house without any involvement of big companies. But I think to produce vaccines on the scale that we produce drugs would need a big drug company behind us, or a big company. And they're not interested in extracting proteins from parasites. They want things they can culture up, sterile, and all the batches are the same. And there's a lot of work to do, but there's progress and promising results of trials that work experimentally. The big challenge is taking them outside and trying them in a field with real sheep that might be infected with other things, and that's where things have kind of got to. So there are projects on for Telider sagea, which would be our #1 parasite in the UK and probably in Ireland too, and Trichostrongylus, some of the come later in the year. We've tried with fluke actually. There's a massive research community working on fluke vaccines. It's the holy grail, it really is. There are some challenges there though, because sheep and cattle do not become immune to fluke. It'll affect them at any time of their life. Unlike worms, older animals are a little bit immune. You don't get that. And you've also got the cells to kind of amplify things, and deer and rabbits to spread it about. And they're more complicated than worms. I'm always teasing my colleagues that fluke are much more complicated. You've got it easy. So we've tried the BarberVax approach actually. I hope I'm not speaking off the record here, with three attempts. And basically the results were equivocal. They weren't good enough. And they're not easy trials to do. They're expensive to run, so we've shelved that for now. We'll let others go on. But there's a huge push on vaccines in Ireland, actually, and fluke vaccines. I'm sure you know Grace Mulcahy. And John Dalton, actually, who's been in Australia and Queens, and I think he's in Galway now. Done some very interesting work on that. But they're looking at the real immunity against fluke. So I think I wouldn't say you'll have a vaccine in a year or two. Bit of a cop out, but we usually say I'll be disappointed if we don't have one in 5 years, and then 5 years comes around and said, yeah, told you, disappointed. It's complicated though. I think, you've seen with COVID, that's just the virus, how difficult it is with, all the effort that's gone into that. Vaccine work against helminth parasites, these worm parasites, is really challenging.
One thing that I do recognize when I talk to scientists and researchers is the level of detail, and people have got an insight into it there, that's fundamental to people on the ground, at farm level, to veterinary practitioners, to anybody involved in it. We don't see that level of detail. We just get the product out and we have a quick look at the label. How does it work? Does it work? Does it work? Then off we go. Are we spending? I know Moredun is probably an exception because it's really ahead of the game on research. Are we putting enough emphasis on research? Because you said, you know, a lot of it has to make a profit with you know, some of the products we produce need to be profitable. Is there enough money going into research? Is there enough people being attracted into that side of the industry? There are two questions, I suppose. Is there enough invested in it, and is there the people coming through with the interest?
Yeah, that's a very good question. I mean, we would always say there's never enough money invested, and we have to compete hard for grant money to do projects like this and to do some of the fundamental research. We're very lucky in Scotland, and I mean, I should have said the Moredun Institute, we're 100 years old this year, and I've been there for a quarter of that. It makes me feel very old. But we were set up by a group of interested and concerned farmers and vets in 1920. And we're now one of the Scottish Government's major research providers. So we get maybe half of our income from the Scottish Government and work with them on strategic things of interest to them. And animal health and welfare is a big one of those. So we're lucky to have that underpinning capacity, if you like, and then compete for the rest. But it's a hard job, to do the science and compete for grants and be competitive in areas that don't always attract funding. I mean, livestock isn't as high a priority as human, and when there are drugs out there, the regulatory bodies think, well, or the farmers think that's covered, and what research do we need? And so it's a really interesting one. I think there are some very talented people, and they're attracted by the technology and molecular biology and computational modeling, epidemiology, all that stuff, and we need to attract good people. And from what I've seen, people coming through are very gifted and talented. I think it's about having the funding and the support and people there for long enough. I mean, I think we're a wee bit different, I guess. I mean, universities are very transient. You have people coming and going all the time, whereas we're a bit more, we have a bit more sort of longevity, if you like, a bit more, projects like Barbavax would never have come to fruition if we'd had a series of short-term research contracts. You just no continuity, you'd never get anywhere. It's because people like David and George and myself, to some extent, were there for 20, 30 years. So that's challenging. So it's really, where does the livestock sector fit? Is it a priority? Who's going to fund it at the end of the day?
It must take incredible discipline. Like I can be creative, but I struggle with long-term focus. That absolute detail. When I see statistics, I always prefer if somebody else breaks down the statistics to one simple map for me. What does it take when you're on a research project that's 5 years or 3 years? And I've seen some people on them for 10 years. God, we all want it next week. What does it take in a person? Is it a certain type of person? Is that something you develop over time?
Yeah, I think so. It depends on the funding sources. I mean, some of them are much more sort of snappy and need things done in a year, or these sort of long-term strategic things. They have to be planned carefully in advance and co-constructed with the people you're going to deliver them with. That can be the pharmaceutical industry or the farmers union or Quality Meat Scotland, or it could be Scottish Natural Heritage, or who you're working with. So they're co-constructed with your stakeholders, and then you engage with them all the way through the process from start to finish. Those longer projects, but it takes a lot of planning. And I mean, I guess in the Scottish Government, for example, we would do that with projects with them over a 5-year period on a sort of rolling research program. But they're all carefully constructed and carefully reported on, and audited and all the rest of it. And it takes that sort of mindset to do that. It's not really my forte, I'd have to say, but some people are very good at it. So it's being very organized and being able to stick with it. And but if you're contracted to do it, then there's an incentive to do that and report progress and all the rest of it. So it takes a certain mindset.
Yeah, no, I definitely see it. I don't have it, but I respect the people who do.
I'm not sure I've got it either.
I know, I think you do. I think it's the work that has been done in research feeds back into the rest of the world. We sometimes, everyone else takes credit for and doesn't get the recognition for the importance. But I think that's the key thing for me there. Okay, let's move aside from parasites just for a moment and to the wider area of livestock and climate change. You have a role currently, I think, with the Scottish Government on an advisory board, is it? Do you want to just talk about that role? Yeah,
it's slightly different to that. Moredun is one of 6 research institutes in Scotland that are funded on this strategic research programme by the government.
Okay.
So that would include organisations like the Royal Botanic Gardens, who have a research wing, and the Rowett Institute in Aberdeen, who do nutrition work and food. You have the James Hutton Institute, very famous environmental research, and Scotland's Rural College. I'm sure you'll know, but they're set up a little bit like Teagasc. They'd be a bit like that. So they've got advisory wings to them, and they teach a bit. And you've also got BIOS, who are biostatistics support, who'd help you sorting out your statistics and for the non-numerate people like me. So they've come together recently in a collective called Safari, and it's spelt wrong. It's spelled SEFARI. So I would encourage people to have a wee look at our website. I guess we are set up a little bit like Teagasc, with the different institutes now under that umbrella. So it's the Scottish Environment, Food and Agriculture Research Institutes, and within that we have what's called the Gateway, which is a kind of knowledge exchange hub, and we have sector leads across that. And there are leads in things like soils and environment and people and communities. And for my sins, I'm livestock sector lead. So that's a knowledge exchange role, really. So my job is to keep an eye on what's going on across the programme. When I'm not an expert in all things livestock, but my job is to keep an eye on people who are. So working across the Hutton and the SRUC on genetics, genomics, breeding, feeding, emissions, all that sort of stuff. I mean, our Moredun's role is very much around endemic disease and reducing the burden of production-limiting disease, as we've described, and that goes way beyond parasites into viruses and bacteria and all sorts. And we're increasing our remit on horses and pigs and poultry and even aquaculture. Okay. So I've got to keep an eye on that, because livestock's not just sheep and cattle, but also help communicate that out to the wider community, and that community can be the public. So public engagement is really important. It's public money at the end of the day. So what are we spending it on? Why are you doing that? Why is that relevant to me? So it's our food, it's our environment. That's and these are core messages to get across. But it's also policy teams who actually make up policy based on science and evidence. That's our job to produce that and discuss it with them and help them understand it and see the benefits of, if you do this, that, or the other policy. And they're big policy areas, like, as we said, biodiversity, climate change. These are global things. And the Scottish Government's very hot on all of those. But it's also the industry. It could be farmers. I mean, Moredun are very well connected with the farming community in the UK, especially. We need to reach to Ireland, I think, as well. I mean, we don't really have much of a base in Northern Ireland, but certainly across GB we do, with regional advisors north to south, telling us what's happening on the ground and what you should be working on and what you shouldn't be working on. So they're important too. And the Farmers Union and the red meat industry, Quality Meat Scotland, people like that. So quite a busy role in that regard, and that's around knowledge exchange. So the website's quite active.
The big question, Philip, though, is, are we getting it right when it comes to, from my experience of seeing some of the science that's happening at the back end and understanding methane cycles and different bits and pieces that are happening, the science is improving, but at times I feel like we're losing the messaging to the general public around livestock. It's a hard one to sell. It can be complicated. We tend to go down to rabbit holes where we're not performing well on, and we don't exemplify the stuff we're doing well. It's very complicated. Are we winning, losing, or somewhere in the middle?
Yeah, that's a really interesting one and very timely. There's a huge discussion now around livestock's role in food systems, both here and across the world, their contribution to climate change. And there's no getting away from that. They do contribute, and we need to measure that and monitor that and quantify that, but also see how we can improve that situation. And it's not, the simple sort of knee-jerk reaction is just get rid of all the livestock, but you have to think of all the collateral damage with that, to the environment, to rural economies, and it's thinking at that sort of level, which I'm sort of fairly new to, but it's a big area, even just in Scotland. I mean, even not looking beyond our border, if you like, if there is such a thing. It's how do you do that sort of thing? And you've all sorts of competing interests. You've people, and agencies promoting biodiversity, and there's others trying to do the opposite. And you see, we want wetland schemes. No, we don't. And it's trying to think, well, what are the win-wins? What are the easy fixes? What are the low-hanging fruit, if you like? What are the things that are unintended consequences. So that's kind of where the research comes in, providing evidence that that's a good idea and that works really well. And there's an added benefit if you do this. But there's no question that livestock have been targeted as you know, the villains of the piece. And I think there's a lot of misinformation around that, and we need to be clear on the facts. And I guess Moredun and Safari can be honest brokers in that discussion, and we try to be. It's a challenge because I mean 80 odd % of Scotland's land cover is what's technically called rough grazing or area, you know, it's less favoured area, whatever, area under natural constraint. So, you know, there are limited uses for that land that would be productive. I mean, there's a big, as you mentioned, big push for rewilding things and planting trees, and you know, so you can have a mixture of those. Can we have agroforestry? Can we have livestock with planting, you know, rather than instead of, and wind farms? And there's a huge debate around all of that, and a lot of interested parties, and we kind of need to be in the mix, providing the evidence and the science and interpreting good and bad practice. I guess that's kind of where we are.
It's a real. Sometimes you get to frustration that facts don't matter to people anymore. With some, you know, particularly when you look at media outlets. And you look at some of the stories that are populist and or maybe just they're cooler, they're easier to digest, they're more radical, particularly around the environment and farming. But so, is there a piece where we had before we went to press record? There we were talking about, you know, the idea of bringing science, which can be very complicated, and bring it to simple messages, to connecting people. Could we do better at that, or is that something that you're falling into the trap of, you know, just, you know, is that something we should be doing, you know, trying to oversimplify things where in fact these things problems are?
Yeah, that's a challenge. I mean, there's an element of dumbing down where it doesn't mean anything anymore, but we do need to simplify science. We're all guilty of our jargon, but so are sheep farmers. They come up with words I've never heard of. And we need to understand each other's language and talk. But I mean, all of this is about people. It's about behaviour change, whether it's a policy change or somebody doing something different in their home, or a farmer doing something different. It's about connecting with people. I think that's kind of core through all of this. You know, you can talk about biodiversity and climate change, but it's people right in the middle of it are going to be the ones enacting it. It's farmers who are going to be the custodians of our landscape. They're the ones who are going to be tasked with doing whatever policy thinks is the right thing, and maybe incentivizing them or helping them to do that, and identifying what might be worth supporting financially, because most of these industries would go to the wall if you know, if they hadn't support. So it's really what's public money spent on, public goods. How do you quantify that? How do you value that? But science has taken a bit of a picking over COVID and various other things, and I think, you know, I'd like to think people trust scientists, but you can see on social media that they don't, and it's very easy to be adversarial about this and start shouting. And I think you need to be very balanced and moderate in our language and deal, you know, pick your battles, because there are some very vocal people out there who don't agree with some of the things that we would do.
But I think someone said to me, and I was in my time in the Farmers Journal, it was, you know, sometimes don't get caught up with the vocal minority. It's the silent majority that we need to talk to, and you're never going to change everyone's behaviour. It's fascinating work. It's great to see the diversity from, I suppose, your career and your interest in parasites, and now to see you involved. And you seem very measured, Philip, in your approach. And it's nice to see you involved in that livestock story, and interesting to be following that. Let's talk a little bit about scientists and then people who've influenced you during your career. What's been your big influences along the way? Be it people, is it people that you worked locally with, or did you have scientists that you admired? Be it ones that went before?
Yeah, that's a good question. Yeah, as I mentioned at the start, I was lucky to have a triumvirate at Queen's who were really inspirational, all of them in different ways. Just on how they talked about the subject, and just so enthusiastic about it, and that kind of intrigued me. And I guess once you get into career sort of mood, it was people that you encounter along the way. And I was very lucky. Stuart Taylor at the Vet Labs was really interested in what we did or what we could do, and those sort of things. You know, just it's gratifying that you're making a difference and people are interested in what you do. It's not just a job. And then you encounter sort of the real big hitters as you go along. There's a very interesting chap called Tim Geary. I don't know if you've heard, but he he worked for a long time in one of the big drug companies and would have been very much involved in the Ivermectin story down the line. Absolute expert, but the nicest guy you could meet and the most fascinating speaker to listen to. He's really funny, but very, very well informed. So Tim Geary's a big influence from a distance. He was the director of the Institute of Parasitology in Montreal, McGill University, and I was lucky enough to get a sabbatical out there. So that kind of moved my interest from the vaccine work to the genetics of drug resistance, because one of his colleagues, a chap called Roger Pritchard, has done a lot of pioneering work on drug resistance to ivermectin in onchocerciasis, river blindness. So it was a chance to work in that lab. And the third person who just happened to be coming through at that time was an Australian guy called Terry Spithill, real fluke expert. So again, another guy to look up online. And they're just the nicest guys and fascinated by what they do. Everybody's opinion matters. Doesn't matter if you're a student or a postdoc or whatever, your opinion's as important as theirs, and they'll challenge you, but you're as important as they are. And those guys, I think, in a career situation were really, really influential. But I guess when it came to Moredun, there would be 2 or 3 colleagues that I worked with. I mean, Dave Knox, really good scientist, pushing the vaccine side of things back in the day. But the head of department, Bob Coop, and another parasitologist, Frank Jackson, were the best 2 I'd ever seen at talking to farmers. I'd never seen that before. I'd only been sort of stuck in a lab and didn't really talk to anybody. And I saw them in action, and it was that kind of thing. And that's changed the direction of my career laterally, I would say. We're trying to follow in their footsteps, and they're big shoes to fill. But it's just the ability to talk to farmers. I'm not from a farming background, so it's trying to learn what works and how to talk to people and engage at a sort of public level, personal level. And I think that's helped, and it's maybe somehow propelled me into this knowledge exchange role in the government Safari Collective. So I guess it's those guys in sequence, really, both locally and globally, would have influenced me over my career.
Brilliant.
I hope I haven't forgotten anybody now, so I'm going to get.
I know. I know there's many things. It's just interesting to see any famous scientists. Before I'm conscious of time now as well. It's like I was reading about Ignace Semelis. I never get his name right. Did you ever hear about that Hungarian guy?
No, not a name familiar to me.
I could have the wrong name now, but he was the guy who discovered washing hands and bedside fever.
Okay. No, I mean, I've never met William Campbell, although I would really like to. I think he would be up there, a Nobel laureate. I mean, that's exceptional. But yeah, I've been lucky enough to engage with guys, and it's the people thing again. It's communicating science, but it's also collaborating to let you do things. And actually, the other person I really should mention, and it'd be really remiss if I didn't, is a very famous Belgian parasitologist called Joseph Verkruijs, who operates out of the vet school in Ghent. They have a big group there. They do fantastic work, but they're incredibly hardworking, very productive, but he's the nicest man you could meet and incredibly hospitable. So it's not just liver fluke that causes liver damage. If you go on a weekend to Ghent with Joseph Verkruijs, you'll have liver damage, I guarantee it. But he works hard, plays hard, really very influential. I mean, none of us could get anywhere near what he's done in his career. So yeah, he'd be another big hitter. It's interesting, it's a kind of contradiction in terms. What's a famous parasitologist look like?
Yeah, no, but it is interesting to see how people, we have different influences that are working around us. And I do get it's funny, everyone's a bit different on these conversations, but it's interesting that conversations need to happen between science and public, that it's all about people, no matter who I talk to, it's about understanding.
So we have to, you forget, at the end of the day, we're scientists, and we communicate our science to each other, through conferences and meetings and project meetings, and that's another level of knowledge exchange. You're exchanging knowledge with your peers and your colleagues. So it's a lot of it's people.
Yeah, it's people. Philip, I really enjoyed this conversation. I would suggest people follow you. You're on Twitter. I'll put the link in the bio as well. In both it's now a video and an audio podcast. So I'll put the link in, and people can follow you. You've put up some interesting pictures and videos again of these parasites that you're fascinating, and you're fascinated by. And I often think we can't see inside the animals. We can't see the damage. And just by visualizing things, sometimes we get a better understanding.
I mean, farmers are even fascinated if you show them what the fluke looks like or the snail that causes it. They say, how on earth does that little thing, knock my cow down? It's as simple as that. And where does it live again? Show me. Those simple things. And it's just it's just communicating on a personal level in language that people understand. I really appreciate the chance to chat, Tommy. You're pretty good at it too, I have to say. Some of the online social media stuff you do is very, very impressive. That's where I saw you first.
No, thanks, Phil. I enjoyed. You're a very humble person. You're very doing a very important role. As I keep saying to farmers, they don't like me saying it, but I think parasites are going to get more complicated. It's not going to be easier. And without people, and I think this is the key thing for me to understand, without research, none of us can do what we do on farm, or none of us can make an impact. So it's nice to get an insight into the detail that's in research, the mindset that's required to pull these projects to. And I wish you a lot of luck in your new role, sort of communication and looking at livestock and climate change and all those roles. And hopefully we will get a pint of the black stuff at some stage at a conference. I've been doing lots of webinars, but there's some of the best networking can be done later.
I have to say, you collaborate and work with people you'd be happy to have a pint with. I think that says a lot.
Yeah.
So be nice to people and work hard. That's kind of a good mantra.
What a great way to end, Philip. You're really appreciate the conversation, and the best of luck, and keep up the good work.
Thank you very much.